before the break. And I will turn it over to Zaf. Go ahead.
Okay, thanks very much, Shannon. Everyone hear me okay, I hope? Right, brilliant. So yeah, thanks, Shannon, for chairing this. Thank you to Serena and Tristan for organising the symposium. And to, I assume, many other people in the background as well, who are doing lots of work to make this happen as well. So first of all, just to say that, so the work that I'm going to describe is about mGluR-dependent plasticity in the hippocampal area CA2. And this work arose through a collaboration with Serena Dudek via a jointly funded PhD student funded by the Wellcome Trust and by NIH. And the student was Massa Samadi. And this work was published in the special issue of Hippocampus recently, references down here. And as you can see, a number of other people listed as authors on this paper also contributed to this work as well. So most of the work, virtually all the work that I'm going to talk about today, is about synaptic plasticity in CA2. Although I'll touch a little bit about some behavioral assays at the end of the talk. So most of the work I'm going to talk about is carried out in slices from 13 to 21-day-old mice, and it involves electrophysiology, where we've done whole cell recordings from CA2 cells and stimulated the Schaffer collateral axons to the CA2 neurons. And in these recordings, we're holding the cells at minus 70 millivolts. And I just want to point out that while most of this response, most of this current is probably being mediated by AMPA receptors upon release of glutamate from the presynaptic terminal, we're not in most of these experiments, unless I say otherwise, we're not actually blocking any of the other ionotropic receptors, of which there are many, or any of the other metabotropic glutamate receptors either. So whilst most of this response is probably AMPA receptor mediated, there may be contribution from other receptors as well. And finally, just to say, we're also not blocking any GABAergic transmission in these experiments either. So while we're holding the cell at minus 70 millivolts, it may still be the case that some of this response might be partly mediated by GABAergic receptors as well as other glutamatergic receptors as well. So if I'll try and call this a postsynaptic current or a postsynaptic response, but if I get colloquial and start calling it an EPSC, then you have to forgive me. Okay, so what we're particularly interested in is mGluR-dependent synaptic plasticity. And of the mGluR types, there are 3 groups, group 1, group 2, and group 3. And we're particularly interested in group 1, which are made up of mGluR1 and mGluR5 receptors. So in the hippocampus, it's been known now for many, many years that if you activate group 1 mGluR receptors with specific agonists, such as this one here, DHPG, which is a selective agonist for group 1 receptors, this produces long-lasting depression of synaptic transmission of Schaffer collateral inputs into CA1. And there's been a lot of work now characterizing LTD, mGluR-LTD in CA1, and we know quite a lot about some of the mechanisms of this form of LTD in CA1. We also know that mGluR-LTD exists in CA3 as well. So this is just one example. So this is now using paired pulse low frequency stimulation rather than a selective mGluR agonist. And paired pulse low frequency stimulation seems to somehow or other release glutamate sufficient to activate group 1 mGluR receptors. So you can see here in the open circles, these experiments carried out in the presence of AP5 or APV, which is NMDA receptor antagonist, to exclude any effect of NMDA receptors here. And you can see under these conditions that paired pulse low frequency stimulation actually produces nice robust LTD. And this is mGluR-LTD because it's blocked by mGlu1 and mGlu5 receptor antagonists, as you can see here in the filled triangles. So we can get mGluR-LTD in CA3, we can get mGluR-LTD in CA1, but we don't actually know very little, if anything, about mGluR, certainly group 1 mGluR-LTD in CA2. There is recent work showing that inhibition of group 3 receptors in CA2 can actually facilitate LTD in CA2, but with regards to group 1 mGluR receptor LTD, which is the most widely studied in other regions, we actually don't know anything about this. So that was the goal of these experiments. Now, there's no reason to think that mGluR receptors aren't present and are not functional in CA2. So if we just concentrate on mGluR1, for example, and mGluR5, They're distributed in CA2, similarly, as far as we can tell from these kind of experiments, similarly in CA3 as in CA1, and the same also for mGluR1 as well. So there seem to be equal distribution of these receptors in CA1, CA2, and CA3. So we would expect these receptors to be doing something in CA2. So in these first experiments, what we've done is simply applied the mGluR, group 1 mGluR agonist DHPG to slices of hippocampus when we're recording from CA2. And those data are shown here in the red circles. So this is normalized PSC amplitude data on the y-axis and time on the x-axis, and DHPG application is shown by the gray bar here. And you can see there's initial depression of transmission, which then is long-lasting for as long as the experiment is carried out. So this is group 1 mGluR LTD in CA2, and it's very robust and very reproducible. So what we did next was just to test whether some of the properties of mGluR LTD that we see in other regions, such as CA1, also apply to CA2. And in this experiment here, we've tested to see whether it relies on protein synthesis. And we've used an inhibitor, anisomycin. This is the data here in green. And you can see that anisomycin, although there's a transient depression, in the presence of anisomycin, the responses come back to baseline. And here's the data, here's the traces from these two sets of experiments. So the representative traces of the control and LTD, and the representative traces from the anisomycin experiment superimposed as well. Now the pooled data is here, which is taken from the last 10 minutes of the experiment here. And you can see there's significant LTD in control conditions, but not in the presence of anisomycin. And there's a significant difference between the control and the anisomycin data. So as far as this property of LTD, mGluR LTD goes in CA2, it seems to be very similar to LTD in other regions, group 1 mGluR LTD in other regions. Then we look to see, well, is there any differences between LTD, mGluR LTD in CA2 compared to the other hippocampal regions? So in the top graph over here, this is group 1 mGluR LTD induced in CA2. This is a completely different set of data in CA2 now. And in a different set of slices, shown in blue, this is recordings from CA1, where we're also applying DHPG. So there's two different sets of slices. And you can see that in contrast to CA2, in CA1, there's very little, if any, LTD under these conditions. And again, you can see from the pool data here, there's significant LTD in CA2. There's no significant LTD in CA1, and there's a difference between CA2 and CA1 in the level of LTD induced by activation of group 1 mGluRs. And we get a similar effect in CA3 as well. So in CA3, again, a different set of CA2 experiments shown here. There's LTD in CA2, but in CA3, again, it's quite variable, but overall, on average, there's no LTD in CA3 as shown in the pool data down here. As you can see, it is fairly variable, but there's no significant overall LTD in CA3, whereas there is in CA2. So this was quite interesting. The fact that we're getting LTD, very significant long-lasting LTD in CA2, but not really in CA1 or CA3. So what are the possible reasons for this? Well, one potential reason comes from this data, which was published actually back in 1990 from the Snyder group. And if we just focus on this panel over here, what this shows is if you activate group 1 mGluRs with an agonist ACPD, and this is looking at tritiated diacylglycerol in hippocampus, in response to activation of group 1 mGluRs with ACPD, you can see this very high level of activation of DAG. So the PI turnover is very high in CA2 in stimulation, in response to stimulation of group 1 mGluR receptors, and it's much lower in the neighboring regions. So it's possible that group 1 mGluRs are more effectively, more efficiently coupled to their downstream signaling cascades in CA2 versus the other regions. And maybe that's what's producing somewhat higher LTD in CA2. But there could be lots of other reasons as well. Now, I don't need to tell everybody here about the vast array of differences in molecular properties of CA2 compared to other regions of the hippocampus. So I'm not going to go through this slide here at all. I'm going to focus in on RGS14. So RGS14, regulator of G protein signaling, is very highly expressed in CA2 compared to the neighboring regions. And we know from previous work that in RGS14 knockouts, you can actually induce LTP in CA2, whereas normally in CA2, you can't, or it's very difficult to induce LTP, whereas the knockout of RGS14 enables LTP. So the question we wanted to ask was, does RGS14 regulate group 1 mGluR-LTD in CA2 in any way? So to do this, what we did was we used RGS14 knockout mice. These are obtained from John Helper. And these data are shown here. So again, in red is a control set, another separate control set of mGluR-LTD induced in CA2. And again, you can see nice, robust LTD. But in the RGS14 knockouts, you can see quite clearly, although there's an initial transient depression upon application of DHPG, there's no lasting sustained LTD. And again, you can see that in the traces over here and in the pool data down here, which is the effect in the last 10 minutes of the experiment. So again, there's no long-lasting depression in the RGS14 knockout animals, and that's the data is significantly different in the wild types versus the RGS14 knockouts. So RGS14 does seem to play some role in regulating group 1 mGluR-LTD in CA2. So given that, we just wanted to know whether, does that apply to other forms of LTD, or is that something specific for G protein coupled forms of LTD, which are activated by group 1 mGluRs? And so we looked at NMDA receptor LTD in the RGS14 knockout animals. So in the top graph over here, these are control wild type animals. And we're using low frequency stimulation. So this is 1 Hz stimulation. And 1 Hz stimulation produces, again, in the red circles, you can see nice, robust, long-lasting LTD. And we just wanted to confirm that this was NMDA receptor dependent. So the green data here is in the presence of the NMDA receptor antagonist AP5, and this blocks the LTD. So low frequency stimulation produces NMDA receptor dependent LTD in CA2. So in the knockout, the RGS14 knockout animals, we then did the same experiment. Low frequency stimulation here again produces nice, robust LTD. This is in the CA2 in the RGS14 knockout animals. So this is the control RGS14 data. And this LTD is also NMDA receptor dependent because it's blocked by AP5. So you can see here that there's actually no difference between the LTD, NMDA receptor dependent LTD that's induced in the RGS14 knockouts versus in the RGS14 wild type. So RGS14 seems to have no effect, no regulation of NMDA receptor dependent LTD, where it does regulate group 1 mGluR receptor dependent LTD. Now, another G protein that's highly expressed, regulatory G protein signaling that's highly expressed in CA2 is RGS4. And we know from previous data that RGS4 does inhibit signaling by group 1 mGluRs. So does RGS4 play a role in mGluR-LTD? And to answer this question, we've used RGS4 knockout animals from the Jackson labs. And you can see from this data here, group 1 mGluR-LTD is pretty much exactly the same in wild type animals versus RGS4 knockout animals. And there's absolutely no difference in these two data sets. So RGS4, in contrast to RGS14, seems to play no role in the regulation of group 1 mGluR-LTD in CA2. So these data, so this is just the data I've already shown you, the RGS14 knockout data and the wild type data. These data were actually a little bit surprising when we first got these results, because as far as we knew, RGS14 actually negatively regulates the signaling of G protein coupled receptors. So we would expect to see an inhibition of mGluR signaling by RGS14. So in a knockout of RGS14, what we might have expected to see, if anything, was actually an enhancement of LTD rather than an inhibition of LTD. So this suggests that there's probably something else going on, some other mechanism that RGS14 is regulating in order to have this effect on LTD, and it might not be through this sort of canonical regulation, negative regulation of G protein signaling. So what kind of mechanisms might there be that RGS14 might be controlling, regulating? One of the proteins we became interested in was this one here, STEP. Now, STEP is striatal enriched phosphatase, and we know from previous work that STEP is important in mGluR-LTD. So this is the work from some years ago now, showing, so what this data here is showing is in wild types and in STEP knockouts, looking at the internalization of GluR1 containing AMP receptors and GluR2 containing receptors in response to the application of DHPG that we know produces LTD. So you can see in the wild type animals, DHPG produces endocytosis of GluR1 and GluR2 containing AMP receptors. But in the knockouts, there's no such effect. So in the knockouts here and here, you can see that there's no internalization of GluR1 or GluR2 containing AMP receptors. So STEP is involved and important for LTD in CA1. What about in CA2? Does it play any role? So we know that CA2 has a high concentration, high density of STEP, and work from Serena's lab, which is part of this paper in Hippocampus, also shows that total STEP, but also active STEP, is very highly localized in CA2 compared to the surrounding regions. So what we did then was to get some STEP knockout animals, and these are from Paul Lombroso's lab, and we used these animals to test the role of STEP in mGluR-LTD in CA2. So as you can see quite clearly here, in the STEP knockouts, there's no LTD whatsoever. Again, there's a small initial transient depression on application of DHPG, but there's no long-lasting depression whatsoever compared to the wild-type controls. And again, you can see the pool data here showing significant depression, LTD in CA2 wild types, but no LTD at all in the STEP knockouts, and a significant difference between the two data sets. So STEP is also playing a crucial role in regulating group 1 mGluR-LTD in CA2. So is there some interaction or some regulation between RGS14 and STEP that might be controlling mGluR-LTD in CA2? So these experiments, again carried out in Serena's lab, these experiments are looking at the levels of active STEP in CA2, in the dendritic regions and pyramidal regions, in the wild type versus in the RGS14 knockout animals. And these immunodata here are quantified in these graphs over here. And you can see this quantification shows that in the RGS14 knockout animals, there's a very large and significant decrease in active STEP in the stratum. So this data is from the stratum radiatum specifically. There's a large decrease in active STEP in the RGS14 knockout animals compared to the wild types. And if you do the same experiment, but look at another CA2 concentrated protein, such as PCP4 here, there's no effect of RGS14 knockout. So it's not that RGS14 knockout has some global effect on proteins specifically in CA2, but there's a specific effect on active STEP in the RGS14 knockout, where there's a decrease in active STEP in CA2. So this might suggest that there's some regulation, we're not quite sure how, but there might be some regulation either of STEP or some other mechanisms that STEP might regulate. And this is the mechanism whereby LTD is controlled via RGS14 and or STEP, rather than being through some G protein signaling that group 1 mGluR receptors might be coupled to. So in order to test this, what we did was to take RGS14 knockout animals and put active STEP back into those cells by infusing STEP into the cells through the patch pipette, through the recording electrode. So that's what this data shows here. So you can see in these gray circles, these are experiments with RGS14 knockout animals where active STEP has been added to the patch pipette. That's been allowed to infuse into the patch pipette, into the cell. And then when you do the experiment, you can see that we get a rescue of LTD under those conditions. And in the other experiments, control experiments with the RGS14 knockout with inactive STEP, there's no LTD. So essentially what's that? And again, you can see the pool data down here. And the STEP peptides, proteins were obtained from Paul Lombroso's lab. So what that's telling us is we have RGS14 knockout and reduced levels of STEP in the RGS14 knockout. By putting active STEP back into the postsynaptic cell, that then allows LTD to be expressed, be induced or expressed. Okay, so the data that I've shown you so far suggests that mGluR-LTD in CA2 has a requirement for activation for RGS14 and activation of STEP. And without these two, then LTD doesn't occur. So in the last little bit of the talk, I'm just going to briefly describe experiments where we're looking at social recognition memory in mice and whether there's a requirement for RGS14 in social recognition memory. So the way that a lot of the experiments are done in mice and in rodents is this kind of experiment that's described here. So this is a social recognition memory experiment where a subject mouse is allowed to interact with either a littermate, so a known mouse, or a novel mouse that it hasn't encountered before. And when you do this experiment in normal mice, what you get is data like this, which shows that these subject mice will spend more time interacting with a novel mouse, which it hasn't come across before, compared to littermate mice, which it has come across before. So this is an indication of social recognition memory. This particular experiment here from Siegelbaum lab, what they did was to silence the CA3 region using tetanus toxin into CA3 region. And what that showed was that these differences in the interaction between the novel and littermate mice actually disappears when you silence CA3. So what that's telling us is that social recognition memory requires some function of CA2 region. This experiment over here, this experiment is investigating the role of STEP in social recognition memory. So it's essentially the same experiment as before, same data, some very similar data to before, where the subject mouse explores the stranger mouse more than it does the known mouse. But in the STEP knockout animals, that effect disappears. So again, we require STEP for social recognition memory. So what we did was did a very similar experiment to test for the role of RGS14 in social recognition memory. So again, we have a subject mouse, and the subject mouse is allowed to interact with either a novel mouse or a familiar mouse, and these subject mice will be wild-type mice or RGS14 mice. And the data that we get is shown here. So if the subject mouse is a wild-type mouse, you can see the heat map showing the interaction is largely with a novel mouse compared to the familiar mouse. And with the RGS14 knockout animals, you can see that this effect starts to decrease. And so there's much more equal interaction with the novel mouse and the familiar mouse in the RGS14 knockout animals. The data is quantified down here as a discrimination ratio. And this shows that in the wild-type mouse, the wild-type mouse is interacting far more with a novel mouse compared to the familiar, whereas in the RGS14 knockout mouse, this effect is very much decreased. So this is telling us that RGS14 is also really important for social recognition memory in mice. So the work that I've described to you has shown that we require RGS14 and STEP for LTD, group 1 mGluR-LTD in CA2, and also that social recognition memory that we know requires CA2 and STEP also requires RGS14. But what we don't know is whether mGluR-LTD itself in CA2 through these mechanisms is involved in social recognition memory. So that'd be an interesting next study, which would be great to be able to do. So that's something that we're looking to try and do in the future. So I just want to say once again, thank you very much to everyone involved in this study, Serena, Massa, Shannon, and various other people who are involved in various ways in making this work happen and getting it published in the special issue of Hippocampus. And also thanks again to the funders, the Wellcome Trust and NIH. Thanks very much for your time and attention, and I'm happy to take any questions.